So thank you again for the invitation, and thank you for setting up this special issue. It was a pleasure to contribute to it. And now I will present to you something about what we do in the field. And the title I chose today is the Glycans as biomarkers, but even more important, as a functional effectors of age-related diseases. Many people still do not know what are glycans, so I'll make a short introduction. So the protein you see revolving in the middle, it's a S glycoprotein or stuff of the COVID virus, and everything hairy on the surface is a glycan. So it's very hard to understand function of any protein because majority of proteins are glycosylated if we ignore glycans. And I like to use analogy of these two birds on the left. This bird, because of a disease, doesn't have a single feather, but it's still a bird. And if you would use this kind of a bird to study birds in general, you could be the best scientist in the world, you can have the best planned and executed experiment, but the flight would be beyond your comprehension. So the closest you could come to understanding flying is understanding this bird walking. This is because bird without its feathers cannot fly, and protein without its glycans cannot function normally. And this is case for many proteins. Maybe the best studied example is immunoglobulin G, where glycans on the Fc domain directly decide to which receptors IgG will bind and what will be the function of IgG. So to understand function of a bird, we need to have feathers, and to understand function of a protein, we have to study its glycans. Before I go on, just a word of caution. The term glycation is way more known. It's something completely different. Glycation, which we quantify as HbA1c or advanced glycation end products, is a random damage. It's taking a shotgun and firing at the protein. It's a chemical reaction between glucose and amino groups, and then the conversion. Glycosylation is a sophisticated modification, which is actually adding additional layer of complexity between the gene sequence and the protein structure. Because the polypeptide part of a protein is encoded in a sequence of nucleotides, and it cannot be changed. It's hardwired for an individual, and actually it's hardwired through generations. While glycans, they do contribute to the structure of a protein, they do contribute to the function, but the exact structure of a glycan which will be added is not encoded in a gene. It's encoded in a network of dozens or hundreds of genes which work together. All the polymorphisms, all the epigenetic regulation, all the environmental factors which affect this network can affect the glycan structure, can affect the protein structure and the protein function. So we should think about alternative glycosylation, so adding different glycans to the same glycosylation site, to be functionally analogous to coding mutation, so the change in the sequence of nucleotides, but it's not inherited in the Mendelian way, it's inherited as a complex trait. And a couple of years ago, we very clearly demonstrated that by generating a collaborative cross cohort of mice, so starting from 8 individuals, crossing them for 3 generations, and then inbreeding the strains, some of the strains became structurally and functionally different from the rest of the strains without a single change in the gene sequence. It's just reshuffling of allelic variants which made these mice different. And we see the same in the human population. So one of the key reasons why we are not all the same, why people are different, is because our glycans are different. And this is just 6 random individuals, different glycan, different color, just to show you by the eye that glycans between individuals vary. The problem with glycans is that they are chemically very complicated. Many glycosylation sites, different structures can be attached to the same glycosylation site. So it is not easy to analyze large cohorts. And we were the one who first went into that direction. We published our first high-throughput glycomic study 15 years ago, more than 15 years ago, and we already analyzed over 200,000 people in collaboration with some of the best researchers all around the world, which enabled us to push glycomics into the more or less into the mainstream of omics, so we can regularly publish in leading journals, including engineering. One of the first things we ask ourselves is, how are how is glycosylation genetically regulated? And through a series of GWAS papers in the last 15 years, we mapped a large network of genes which regulate IgG glycosylation. And for now, for only IgG, we have over 40 genes. Only few of them are enzymes which actually create the glycan, and most of them are the regulatory genes which decide which kind of glycan will be added. And this is something what is protein specific, because when we did the parallel GWAS of transferrin and IgG, which have a very similar glycan structure. So the glycans attached to two different proteins are more or less the same. Enzymes which make them are more or less the same, but the regulatory network is completely different. So genes which regulate glycosylation of transferrin are completely different from genes which regulate glycosylation of IgG. And these genes are also known risk factors for different diseases. So we have shown that the genes which regulate IgG glycosylation are highly pleiotropic with at least 94 different phenotypes, starting from expected one like a lupus or type 1 diabetes, but also unexpected one like a Parkinson's, some forms of dementia, or HDL concentration. Another thing we realized very early is that glycans change a lot with age. So this is a relatively recent paper, 5 large populations, different glycans, and how they change with the age of an individual. And based on this knowledge, we developed something we now call the glycan age clock of biological age, which is a commercially available test which measures composition of the IgG glycan. And we know that one type of structures are mostly found in older, unhealthy individuals, while the other type of structures are mostly associated with young and healthy individuals. And we know there are numerous environmental factors which affect this. And most importantly, these glycans are functional effectors of inflammation. They either promote or suppress low-grade systemic inflammation. And of course, I have to repeat my disclaimer here. I'm a co-founder and shareholder of Glycan Age, so obviously I'm conflicted. So please make your own conclusions, and I will just present the data. So one thing which we know from study of a large number of people all around the world is that glycans are functional effectors of inflammation. So they have multiple roles in balancing inflammation. They're not only biomarkers, they're effectors of inflammation. And also we know that they integrate genetic information, so something what is written in our genome, epigenetic information, so the knowledge acquired from the moment we were an egg in our grandmother, and environmental factors which all integrate together and decide about the glycan composition. Approximately 40% of glycan age is heritable, while the rest is affected by lifestyle. And we also know that the changes in IgG glycan happen much sooner than the disease develops. So actually, these molecules are drivers which drive from the healthy condition into some kind of a disease linked with a chronic inflammation. And these are more or less all diseases which are burdening society today. And we studied dozens of different diseases, and in the majority of them, we saw the changes which go in the direction of aging. So a healthy individual looks younger compared to an individual with many different diseases, which have a glycome resembling a much older healthy individual. And these changes happen up to a decade before people develop disease. And we believe this is actually a measure of underlying low-grade chronic inflammation. And one interesting example was a COVID. And this is one of the papers we published in this special issue of Engineering, where we found that the acceleration of glycan aging, and some people with a severe COVID would gain 10, 15 years within a couple of weeks, was actually a predictor of fatal outcome. So people who aged rapidly with a severe COVID, they most likely will die. Another example where we think IgG glycan is very revealing are the cardiovascular diseases. And the first large study we did in this direction was together with Tim Spector and Jim Wilson in UK. We analyzed over 3,500 people, and we saw that the IgG glycan composition associate with the cardiovascular disease risk score. So they're kind of associating with the risk of future cardiovascular diseases. And based on that data, we got the samples from the EPI cohort, the German part of EPI cohort. This is 27,000 individuals with samples collected over 30 years ago. And during the follow-up time, many people had either heart attack or stroke or developed diabetes and so on. And when we looked at the glycans at the baseline, only the IgG glycan composition was as predictive as entire AHA score in men, and even more predictive than the AHA score in women. This was published 2 years ago, but a single finding is never convincing. So we did a replication study, which was published last year, where we did show again that the glycans predict heart attack and stroke, and they do predict it in a sex-specific manner. So different glycans associate with the risk in men and women. And then we went for another 2 replications. This is still not published. It should be published soon in Circulation Research, where we showed exactly the same associations in 2 additional cohorts. So we can be pretty confident that the IgG glycan composition really predicts future cardiovascular events. And there are additional evidence that they actually may be causative, because if you kind of trick this glycan age clock of aging, meaning you improve the cellulation by feeding the mice with a precursor for cellulation, which is MANAC, you can actually protect obese mice from developing hypertension. So we don't have a data in humans yet, but in mice, this seems to work. So the question for humans is, can we revert this glycan age clock of aging? So can we make a decision and slow it down, and in this way revert inflammation? We know from a paper published a couple of months ago that there are at least 16 genetic loci which are associating with the glycan age clock of biological age. So there are many potential candidate genes which can be targeted to affect it. And we know that there are some drugs which are pretty effective. For example, anti-inflammatory biologicals like infliximab or adalimumab, they can really reduce the IgG, the glycan age of IgG, or these pro-inflammatory IgG glycans. But this is not something you want to do preventively. We also had recently a placebo-controlled randomized trial of metformin, and there we learned that despite the anecdotal evidence that metformin is actually working, it doesn't seem to work, at least in the majority of people. So this is a placebo, this is a metformin branch, and there was only actually a single individual who responded well to metformin. We know that hormones are very important. So when we look at glycans during the transition from premenopause to menopause, we see a very large acceleration of glycan aging in women. So the blue are men, red are women. This is a perimenopause period. And in principle, what we see that the women will have a much slower pace of aging than the rapid acceleration during the perimenopause, and then slowing down again. And again, to verify it, we did a placebo-controlled randomized trial with the blockade of the natural hormones, and then the supplementation with either estrogen in women or testosterone in men. And indeed, estrogen is both sufficient and necessary for the kind of youthful IgG glycan, meaning for these anti-inflammatory glycans. So when we looked at the individual level, we know that the perimenopause is kind of associated with the rapid changes in hormone levels. So measuring hormones doesn't really help much. And IgG glycan could be kind of a tool to integrate these changes. And we do hope that IgG glycan profiling for estimating estrogen concentration will be used in a similar way as HbA1c is used for the average levels of blood glucose in the past weeks. Another paper we published in this special issue is the effect of regular menstrual cycle on the IgG glycan composition. And indeed, we do see variation of IgG glycan during the menstrual cycle of a single individual, but these variations are relatively low at the level of few percent of a glycan. So in a cohort study, this is not an issue. Another way to change your IgG glycan is to lose weight. So bariatric surgery is extremely effective. We had a patient who lost 37 glycan years in a few months. Of course, bariatric surgery may be too drastic for too many people, but even the regular weight loss, and this is 2,000 people which we tracked for 15 years, some of them were gaining weight, some of them were losing weight, and those who were gaining weight were aging faster than those who were losing weight, and this was happening in any period of life, so in the 30s, in the 40s, in the 50s. If you start losing weight, it's beneficial for your IgG glycom composition. We had 1,000 people on a low-calorie diet for 2 months. They all changed, the IgG glycom changed, so low-calorie diet is working, but low-calorie diet is not sustainable. So after 2 months of being hungry, these people were randomized to 5 different diets. And interestingly, each of the diets was beneficial for some individuals and not beneficial for other individuals. So I think the conclusion here is one size fits all does not work. There is no magic diet. Each of us has to find a magic diet for ourselves. The same goes for physical activity. In principle, physical activity is beneficial, but too much physical activity is not. So we also see people who exercise too much to have a negative effect. Microbiome, extremely important. If you do fecal microbiota transplantation, your glycom will change, but this also requires a lot of work. So glycan clock of aging can be reverted. We have many individuals making a decision. This Tim Spector, famous researcher, and then he lost 11 years in 6 months after changing lifestyle. He even recorded Instagram Live on that. And the big picture is that if we make this kind of a decision soon, before we become ill, maybe we can prevent disease or at least postpone it. And we all know that we need to maintain our cars. We will not change our tires only after they explode on the highway. But unfortunately, this is what we do to our health. And hopefully this will change. And at the very end, I have technology, have a very large team of people, nearly 50 at the moment. And we got a lot of funding from a different funding agencies, which enabled us to do all this work. And I thank you all for your attention, and more than happy to answer questions.
工程创造人类美好未来。